3 of genome annotation. Module 3 is the cellular localization data. This module predicts where in your cell the protein localizes. So what you do is you basically scroll through your lab notebook. So let's go back to the top. So the first module we did was the basic information module, which gave us some basic information about the DNA and the protein of our gene. And next we did sequence-based similarity data, where we pulled up proteins with similar sequences to our protein. So we used blast search, and then we used T-coffee to align our 11 sequences. And then finally, what we did was we made a web logo, which told us where the conserved regions or conserved domains in our protein was located. So the bigger the letter, the higher the conservation among these 11 sequences. Now today we are going to look at the cellular localization of this protein. So the first thing is basically to figure out whether this organism is gram positive or gram negative. You can look that up by going directly to PubMed. You can also Google it. I know for a fact that the gram stain of this archaea is negative. So this is a gram negative archaea. It might be different for your organism, so please do go and Google it. Then the next thing we're going to actually do is the phobias. We're doing phobias because TMHMM is currently offline for some reason. So basically you control click, open up a new window and go to phobias. What phobias will predict is basically where your protein is located in the cell. So for that, you need your protein sequence. So go back to your lab notebook. So scroll up top and you will see your protein sequence. So here we go. Here's the protein sequence. Then you copy and paste your protein sequence into phobias. Okay, we've pasted it. Select your output format. It should be long with graphics. And then hit submit. Okay, so what phobias should give you is a graph. Okay, let's go back to my original phobias graph in my annotation. So what you do is you copy paste all the information and also the graph from phobias. Okay, so this is my phobias graph. The transmembrane regions are in gray. So these are the regions of your protein which cross the plasma membrane, or actually the membrane. It doesn't have to be the plasma membrane. It could be mitochondrial membrane, so on and so forth. So these regions of your protein are transmembrane. The blue regions are non-cytoplasmic. The green regions are cytoplasmic. And if you have any red, that will locate your signal peptide. So I have two transmembrane regions in my protein. So if you look at the information that phobias gives you, it tells you that you have a signal sequence which spans from amino acid 1 through 27. So signal sequences basically tell the protein where to go in the cell. Then it tells you your transmembrane region, so transmembrane region. It also tells you your cytoplasmic and non-cytoplasmic regions. So basically what my protein has is two transmembrane domains and cytoplasmic regions and also extracellular regions. So it looks like if my protein is spanning the membrane twice. Now you do exactly the same thing with p-sort here and Phobius. And once you've pasted in your amino acid sequence into p-sort and into Phobius, what it will give you back is some data. It will give you localization scores. So the higher your localization score, the higher the probability that your protein is present in that cellular compartment. So from p-sort, I can see my highest p-sort score is in the cytoplasmic membrane. So p-sort is predicting that this protein is in the cytoplasmic membrane. Okay. Phobius also predicted that my protein had transmembrane helices, suggesting that it is in the plasma membrane. If you look at TMHMM, so basically you copy paste this and it will open another tab, and then what TMHMM tells me is that the number of transmembrane helices is 2. So this is the length of my protein. This is the number of transmembrane helices, the number of amino acids in my transmembrane helices, so on and so forth. So if you look at this, sequences 1 through 8 are inside the cell. Amino acids 9 through 28 are part of the transmembrane helix. Amino acids 29 through 1640 are also are in the extracellular region. Amino acids 1641 through 1663 are in the transmembrane region, and the rest of the amino acids are in the cytoplasm. So when you write in your conclusions at the end from all 4 of these software, so TMHMM, p-sort, Phobius. Okay, there were 3. So it's TMHMM, p-sort, and Phobius. So you'll be asked to put in your hypothesis. So based on the results of these tools, where do you expect to find the protein? So my hypothesis is that this protein is located in the cellular membrane. So it is probably a membrane protein because it contains transmembrane helices. So the next module, so now what we've done so far is found similar proteins to our protein of interest, and we've also figured out that it is probably a transmembrane protein. The next module that we are going to do is the structure-based evidence module, which is a part of the next screencast.